Beta oxidation of fatty acid occur in the mitochondria. It is a catabolic pathway, and if you draw a fatty acid, we have functional group that is the carboxylic acid at carbon number 1. Then we have like this number of carbon atom. The carbon that is attached to the functional group is known as the alpha carbon, and the adjacent is known as beta carbon. So second carbon number is the alpha carbon if the functional group is marked as 1, and second is the alpha, third is beta, like that. The last one is omega carbon. So beta oxidation of fatty acid, that means oxidation of the fatty acid occurs at the beta carbon atom. That means this is where the cleavage occurs at the carbon atom between 2 and carbon atom between 3. So alpha and beta, between alpha and beta. This is the beta oxidation of fatty acid. The product which you get is acetyl-CoA. So you get acetyl-CoA, that is a product of the cleavage of fatty acid at oxidation of beta group. So at the beta oxidation you get acetyl-CoA, and this cleavage is catabolic reaction. It is a catabolic pathway, so it occurs in the mitochondria, and it is activated by glucagon and it is inhibited by insulin. This we have already covered at the concept. So the first reaction that occurs at beta oxidation is fatty acid is broken down and we get acyl-CoA. So CoA is a high energy bond. Now what is the rule of business? Rule of business is if you want to get energy out of something, you have to invest some energy, right? So this is the rule of business. So for the activation, for the breakdown of fatty acid, you need to put energy first. So ATP is used. How many ATP are used? 2 ATP are used. So ATP give rise to AMP, monophosphate. Triphosphate gives monophosphate. So you use 2 ATP for this reaction. Now the end product, what is synthesizing in this reaction is acyl-CoA. So the enzyme is known as acyl-CoA synthase, which is also known as thiokinase, which has a EC number 6. Now this reaction occurs in the cytoplasm, and then we have the rest of beta oxidation occurring at mitochondria. So the product which we get in the first step is fatty acid is broken down to acyl-CoA, right? And this reaction occurs in the presence of enzyme acetyl-CoA synthetase, which is also known as thiokinase. This reaction occurs in cytoplasm. 2 ATP are used. Now you have to remember this, 2 ATP are used in this step. So when we talk about the energetics of beta oxidation, we have to subtract these 2 ATP because these are used first. And then we calculate the net ATP which we get out of the beta oxidation. So these ATP are used first. You have to remember in the first step we have used 2 ATP. Now fatty acid gives rise to acyl-CoA in the cytoplasm. This acyl-CoA goes into mitochondria for further beta oxidation of fatty acid, and we have 3 enzymes. In the presence of carnitine protein, there are 3 enzymes that are used in this reaction for transportation till the mitochondria. First one is CPT1. CPT is carnitine palmitol transferase. This enzyme is the rate limiting enzyme of beta oxidation of fatty acid, which is carnitine palmitol transferase. This is present in the outer mitochondrial membrane. In the inner mitochondrial membrane, we have 2 enzymes. Another one is CPT2 and translocase enzyme. Now CPT1, which is present on the outer mitochondrial membrane, this is the rate limiting enzyme. So this CPT1, which is carnitine palmitol transferase 1, this decide the entry of acyl-CoA into the mitochondria. So this acyl-CoA group, when it reaches to mitochondria, it requires CPT1 enzyme, and this enzyme is required for very long chain fatty acids. So for long chain fatty acid from 14 carbon atom to 20 carbon atom, we require this carnitine system. And for medium chain or short chain, medium chain such as 6 to 12 carbon and short chain such as 2 carbons, fatty acid can directly enter into the mitochondria. So this is the significance of carnitine palmitol transferase 1 enzyme, which is the rate limiting enzyme. Now this acyl-CoA from cytoplasm, it reaches to the mitochondria. Another system which we have studied earlier is fatty acid synthesis, in which acetyl-CoA forms malonyl-CoA, and this reaction, if malonyl-CoA is produced, it inhibits CPT1 enzyme. So this means that CPT1 enzyme is the rate limiting enzyme. So this means if fatty acid synthesis occur, beta oxidation of fatty acid cannot occur because malonyl-CoA, which is a product of fatty acid synthesis, it inhibits CPT1, therefore it inhibits the beta oxidation of fatty acid. So if fatty acids are synthesizing in the body, there cannot be oxidation of fatty acid occurring at the same time. So in case of a fed state, wherein the fatty acid synthesis is occurring, beta oxidation cannot occur. And in case of starvation and fasting, when beta oxidation of fatty acid occur, in that case malonyl is not formed, no inhibition of CPT1. So this regulation is known as reciprocal regulation because these two processes cannot occur simultaneously. So if you have a look at the structure of a fatty acid, long chain fatty acid, in the mitochondria of the liver, this is the structure in which one end we have carboxylic end, another end is the methyl end. So this is the fatty acid. Cleavage occurs at the beta between carbon atom 2, carbon atom 3. First is the carbon atom which is functional group. Second carbon, which is the carbon that is adjacent to the functional group, is known as alpha carbon. The carbon which is adjacent to the alpha carbon is known as beta carbon, and the last carbon to which methyl group is attached is known as omega carbon. So cleavage occurs between carbon atom 2, carbon atom 3, which is cleavage occurs at the alpha carbon atom. So this cleavage gives a product which is acetyl-CoA. So how many acetyl-CoA will we get? 1 acetyl-CoA, 2 acetyl-CoA, 3 acetyl-CoA, 4 acetyl-CoA. First, let's see the cleavages that occurs. So these are the cleavages we get. How many cleavages? 1 cleavage, 2 cleavage, 3, 4, 5, 6 and 7 cleavages occur. And how many acetyl-CoA we get? 1 acetyl-CoA, 2 acetyl-CoA, 3 acetyl-CoA, 4 acetyl-CoA, 5 acetyl-CoA, 6 acetyl-CoA, 7 acetyl-CoA, and we get 8 acetyl-CoA. So as a result of beta oxidation of fatty acid, which is a 16 carbon atom, for example, we take palmitic acid, we get 8 acetyl-CoA in the 7 cleavages. So 7 cleavages we get 8 acetyl-CoA in cleavage of 16 carbon palmitic acid when it reaches the mitochondria of the liver. So for each cleavage, we need 4 enzymes. Dehydrogenase, which makes FADH2, and 1 FADH2 gives us 1.5 ATP. So in one cleavage, we require these 4 enzymes: dehydrogenase, hydratase, dehydrogenase and thiolase. Next, dehydrogenase make NADH. 1 NADH gives us 2.5 ATP. So in one cleavage, we make 4 ATPs. And how many cleavage we get from 116 carbon fatty acid, which is palmitic acid? We get 7 cleavages. So total of 7 cleavage and 8 acetyl-CoA we get. So acetyl-CoA, let's talk about energetics first. If we take a 16 carbon fatty acid, which is palmitic acid, we get 7 cleavages. In one cleavage, these are the cleavages, and in one cleavage we get 4 ATP. So in 7 cleavages, we get 7 into 4, that is 28 ATPs. We get only from the cleavage. Now we also get acetyl-CoA. This acetyl-CoA, when it goes to TCA cycle, it gives 1 acetyl-CoA in the TCA cycle gives 10 ATPs, and we have 8 acetyl-CoA molecule. So 1 acetyl-CoA in the Crab or TCA cycle gives us 10 ATPs, and 8 molecules of acetyl-CoA gives us 10 into 8 ATP, that is 80 ATPs. So from 7 cleavages and 8 acetyl-CoA, we get 80 plus 28 ATP, that is 108 ATPs. But you have to remember, in the first step, during the breakdown of fatty acid, we get acyl-CoA. So this reaction occurs into cytoplasm, and this reaction requires 2 ATP. So we have to subtract 2 ATP. For the cleavage of a 16 carbon fatty acid, we get 106 ATPs. So easy way to remember is for a 16 carbon fatty acid, we get 106 ATPs. Energetics for a 18 carbon fatty acid, that is stearic acid, we get 120 ATPs. Now this is if the acetyl-CoA goes to TCA cycle. In previous lectures we have also covered acetyl-CoA can be used for fatty acid synthesis. Acetyl-CoA can also be used for activation of gluconeogenesis. So first preference for this acetyl-CoA goes to the TCA cycle. Second preference of acetyl-CoA is fatty acid synthesis, and the third preference is activation of gluconeogenesis. So if you get question like if the end product of beta oxidation is acetoacetate, then how many ATPs are formed? Now acetoacetate is a primary ketone body, which we have covered already in the ketone body synthesis. So if the end product of beta oxidation is a ketone body, that means this acetyl-CoA is not going to the TCA cycle. It is going for the ketone body synthesis. So the cleavage of a 16 carbon palmitic acid is 7 cleavages and 8 acetyl-CoA. Now from 7 cleavages, in one cleavage we get 4 ATP. So for 7 cleavages we get 28 ATPs. And one acetyl-CoA, when it reaches the TCA cycle, it gives 10 ATP. But here acetyl-CoA is not going to the TCA cycle. So this ATPs we are not getting. So we are only getting 28 ATPs if the acetyl-CoA goes for ketone body synthesis. Now 2 ATPs are required for the activation of the fatty acid, that is from conversion of fatty acid to acyl-CoA. This reaction occurs into the cytoplasm and it requires 2 ATPs. So we get 26 ATPs if a fatty acid goes to beta oxidation, if the end product is, if the fatty acid goes to the ketone body synthesis. So if you are getting a ketone body as the end product, then the ATP which we are getting are only from the cleavages, not from the TCA cycle. So this is how you can solve the multiple choice question. Now coming to dehydrogenases. So as we have talked about dehydrogenases, out of 4 enzymes are the dehydrogenases. We have 2 kinds. One is the long chain dehydrogenases, another one is the medium chain dehydrogenases. Long chain dehydrogenases breaks the long chain fatty acid and it breaks and leaves 12 carbon atom. So it breaks until the fatty acid reaches up to 12 carbon atom. Medium chain acyl-CoA dehydrogenase breaks the fatty acid when the fatty acid is less than 12 carbon atom. So it breaks below the 12 carbon atom. And MCAD, which is the medium chain acyl-CoA dehydrogenase, its deficiency can lead to hypoglycemia and low ketone bodies. So what happens in the normal state, fed state, as beta oxidation is not required in the fed state, only during the fasting and starvation is when the beta oxidation of fatty acid occurs. So what happens in case of MCAD deficiency, since the very low amount of acetyl-CoA is formed, if very low amount of acetyl-CoA is formed, this acetyl-CoA goes only for TCA cycle. This acetyl-CoA does not use for ketone body synthesis. This acetyl-CoA is not used for the gluconeogenesis. Now, since there is a deficiency of this acyl-CoA dehydrogenase enzyme, if there is a deficiency of this enzyme, that means long chain fatty acids are not breaking down. And if this deficiency is, if this is a long chain fatty acid, so there is a breakdown up till 12 carbon atom. After this 12 carbon atom, MCAD, which is medium chain acyl-CoA dehydrogenase, works. But here, if there is a deficiency of this MCAD enzyme, this 12 carbon on less than 12 carbon fatty acid is not breaking down. So there is more of this 12 carbon fatty acids in the body, first thing. Another thing is that if this fatty acid is not breaking down properly, we get only very less cleavages and less acetyl-CoA. If we are getting less acetyl-CoA, the acetyl-CoA which we are getting are only going for the vital cycle, that is the TCA cycle. Acetyl-CoA is not going for ketone body synthesis. Acetyl-CoA is not going for activation of gluconeogenesis. If this is not occurring, if the ketone body synthesis is not occurring, the patient goes to hypoglycemia. If gluconeogenesis is not if the ketone body synthesis is not occurring, then the patient goes to low ketone bodies, because ketone bodies is not synthesizing even in the starvation. And if gluconeogenesis is not occurring, then the patient goes to hypoglycemia. So we get symptoms like hypoglycemia and low ketone bodies. Apart from that, 12 carbon atom fatty acids is also increasing. In this case, we are getting omega oxidation of fatty acid. So what is the major oxidation of fatty acid? It is the beta oxidation. What are the minor pathways of oxidation of fatty acid? It is omega oxidation and alpha oxidation. Omega oxidation of fatty acid occurs at the omega group. So first carboxylic end is the functional group, then we have alpha, then beta. Like this we have the last carbon atom, that is the omega carbon atom. And if this carbon atom is oxidized, it gives dicarboxylic acid. And since there is a omega oxidation occurring in case of deficiency of MCAD, therefore dicarboxylic acid is increased and the patient are seen with dicarboxylic acidosis, that is because of the omega oxidation. Now alpha oxidation of fatty acid occur in the peroxisomes and it is for branched chain fatty acid. Alpha oxidation occurs for branched chain fatty acid and this occurs in the peroxisomes. Now we have a disease which is Refsum disease. In the Refsum disease, this phytanic acid is deposited. Phytanic acid is deposited. Phytanic acid is present in the dairy products and dark green leafy vegetables. So phytanic acid, which is present in the dairy product, in Refsum disease, this phytanic acid is deposited due to defect in the alpha oxidation, and this phytanic acid is deposited in the peroxisomes. So for these individuals who are having Refsum disease, in these individuals we remove the dairy product and dark green leafy vegetable. Oxidation of very long chain fatty acid, this is when it is more than 20 carbon atom, and this oxidation occur in peroxisome. Alpha oxidation also occurs in peroxisome. Another syndrome which we have is the Zellweger syndrome. This is most severe peroxisomal biogenesis disorder. All the enzymes in peroxisomes are absent in this disease. So peroxisome becomes empty, and that is why we call it ghost peroxisomes, because the peroxisomes are empty, there are no enzyme. Since there are no enzyme, no alpha oxidation occur. If no alpha oxidation occur, there is increase in the phytanic acid, similar to that of Refsum disease, which we have studied. Along with that, there is increase in very long chain fatty acid. Why very long chain fatty acid? Because oxidation of very long chain fatty acid occur in peroxisome and there are no enzymes in the peroxisome. So therefore increase in the very long chain fatty acid is also seen in Zellweger syndrome. Now coming to odd chain fatty acid. So odd chain fatty acid means, even chain means there is a cleavage. We get acetyl-CoA in the end, and this acetyl-CoA either reaches to the TCA cycle, or it can be used for beta ketone body synthesis, or it can also be used for activation of gluconeogenesis. But what if there are odd chain fatty acid? Then what we get is acetyl-CoA molecules. These acetyl-CoA molecules are 2 carbon. Apart from that, we get 1,3 carbon compound, that is propionyl-CoA. This propionyl-CoA, which is a 3 carbon compound, we get in the end. So this acetyl-CoA, the molecules of acetyl-CoA goes to TCA cycle. In one TCA cycle, one acetyl-CoA gives 10 ATPs. How many ever carbon molecules are there, we get accordingly acetyl-CoA during the cleavages. But the last product which we get is a 3 carbon compound, because we are talking about odd chain. So say suppose instead of 16 carbon atom, we have 15 carbon atom. So cleavages occur. So suppose if we take odd chain fatty acid, cleavages occur at this, like this, we get 1 acetyl-CoA. another acetyl-CoA, and we get a 3-carbon compound in the end, which is propionyl-CoA. This propionyl-CoA is converted to D-methyl-malonyl-CoA. This is a 4-carbon compound. Since carbon dioxide is added, the enzyme is carboxylase enzyme. This carboxylase enzyme uses vitamin B7. So in case of vitamin B7 deficiency, this reaction is not occurring and there is deposition of propionyl-CoA in the urine. Right. Now the next reaction is from D-methyl-malonyl-CoA, we get L-methyl-malonyl-CoA, that the enzyme is racemase enzyme. So the 4-carbon how we are getting is 1 carbon is the methyl, 3 carbons are malonyl-CoA, so total of 4 carbon we get. Now here also L-methyl-malonyl is also a 4-carbon compound, and from L-methyl-malonyl-CoA we get succinyl-CoA with the enzyme methyl-malonyl-CoA mutase, and this enzyme require vitamin B12. If there is deficiency of vitamin B12, then succinyl-CoA is not formed. There is deposition or presence of L-methyl-malonyl-CoA in the body or in the urine. Now from L-methyl-malonyl-CoA, succinyl-CoA is formed, and this succinyl-CoA is an intermediate of TCA cycle. It enters the TCA cycles and gives us the ATPs. So this is about the odd chain fatty acid. Now coming to cholesterol. Cholesterol is formed by steroid, which is present in the nucleus, and one or more than one alcohol group, which is OH group. Now, if you see the structure of cholesterol, it is cyclo-pento-per-hydrophenanthrin ring. So, if you see the structure of cholesterol, there are 3 hexagonal rings and 1 penta ring. So, A, B, C and D, and to this penta ring, one group is attached. At the first ring, 3rd carbon atom, one OH group, alcohol group is attached. So OH group is attached at the 3rd position, and the cholesterol structure is C27H45OH. So cholesterol is useful for body. It is not bad. It is used for bile acid synthesis, to maintain the membrane fluidity, for vitamin D synthesis, or for steroid hormone synthesis. Synthesis of cholesterol mainly occurs in the liver. Apart from that, cholesterol synthesis occurs in adipose tissue, adrenal cortex, skin, gonads and intestine. Now, if you see the steps in the cholesterol synthesis, if you remember ketone body synthesis, ketone body synthesis is 2 molecules of acetyl-CoA gives 1 molecule of acetoacetyl-CoA, then HMG-CoA and acetoacetate. This is the primary ketone body. First ketone body that is formed is acetoacetate. These are the steps in the ketone body synthesis. It occurs in mitochondria. Because it is a catabolic pathway, it occurs in mitochondria, right? Now the steps are same in mitochondria as well as in cytoplasm. So the similar steps like ketone body synthesis we see in the cholesterol synthesis. So initial steps are similar till HMG-CoA. 2-acetyl-CoA gives 1 acetoacetyl-CoA, which is a 4-carbon compound in the presence of enzyme thiolase. This we have covered in the ketone body synthesis also. But this is occurring in cytoplasm. Cholesterol synthesis occurs in the cytoplasm. Then from acetoacetyl-CoA we get HMG-CoA in the presence of enzyme HMG-CoA synthetase. Then from HMG-CoA we get mevalonate. So mevalonate, which is a 6-carbon compound, we get from HMG-CoA, and the enzyme is very important. This is rate limiting enzyme in cholesterol synthesis, which is HMG-CoA reductase, and this step is inhibited if you give statins, bile acid or cholesterol to the patient. So this is how you can reduce cholesterol synthesis by giving drug like statin. It inhibits the HMG-CoA reductase and therefore it inhibits the cholesterol synthesis, and that is how statins work. So from mevalonate we get a product that is known as farnesyl pyrophosphate. Farnesyl pyrophosphate we get from mevalonate, and this farnesyl pyrophosphate is a 15-carbon compound and it is used for synthesis of coenzyme Q. Coenzyme Q is very important because it is used in the ETC cycle and it gives energy. So from farnesyl pyrophosphate, we get squalene. Squalene, which is a 30 carbon compound. Now we get lanosterol and then we get cholesterol. Lanosterol, which is also a 30 carbon compound, but it is the first cyclic compound. We know cholesterol is a cyclic structure. It is not a linear structure. It is a cyclic structure. The first cyclic structure we get in cholesterol synthesis is lanosterol. Lanosterol and cholesterol, this occurs in the smooth endoplasmic reticulum. So cholesterol synthesis is an anabolic pathway because it is a synthesis. So it occurs in the cytoplasm. Insulin activates the synthesis and it increases the HMG-CoA formation. While on the other hand, in mitochondria, insulin inactivates and it decreases HMG-CoA formation. So CoA, which is a high energy bond, and if they ask question like insulin increases HMG-CoA where? It increases HMG-CoA in the cytoplasm. Statins, the action of statin is, you have to remember that it acts on inhibiting HMG-CoA reductase. HMG-CoA reductase is the rate limiting enzyme in case of cholesterol synthesis. Now you get question, a clinical based question like a patient that is taking statins, patient receives red color urine, and the reason of getting this complaining the red color urine is that the patient who are taking statins, so the normal reaction is mevalonate, which is a 6 carbon compound, forms farnesyl pyrophosphate. This farnesyl pyrophosphate, which is a 15 carbon compound, and the reaction here is the enzyme above is our HMG-CoA reductase, right? HMG-CoA reductase. This HMG-CoA reductase, which is converting from HMG-CoA to mevalonate, this reaction is not occurring because HMG-CoA reductase is inhibited by the statins. Since statin inhibits HMG-CoA reductase, no mevalonate is formed. If no mevalonate is formed, no farnesyl pyrophosphate is formed, and farnesyl pyrophosphate is important or it is used for coenzyme Q. Since coenzyme Q is used for ETC cycle and there is no energy that is coming from ETC cycle, so in the patient who are taking statins, if they exercise much, then the muscle cells damage and they releases myoglobin. This myoglobin comes in the urine and therefore the red color is produced in the patient who are taking statins. Now when you are prescribing statins to individual, those individuals are also given coenzyme Q. Coenzyme Q is also present in the antioxidant capsules so that the myoglobin breakdown does not occur. So statins are given along with the coenzyme Q.